Jai Hind, all my dear students. How are you? I hope you all are fine, right? So, in the preceding sections, we discussed about the structure of embryo, right? So, today we are going to learn the differences between a dicot embryo and a monocot embryo. We have already discussed the structure of a dicot embryo and a monocot embryo. So, today we are going to learn the differences. The first is your number of cotyledons, right? Number of cotyledons. There are two cotyledons attached to the embryonal axis, right? कितने cotyledons होंगे यहाँ पर? दो cotyledons हमें देखने को मिलेंगे. Then in case of monocot embryo, only one cotyledon attached to the embryonal axis, right? Here the position of plumule, right? Here the position of plumule, it is occurs distally, right? Distal position पर होगी. And in case of monocot embryo, it is in lateral position, right? Lateral position. Okay. Now the position of cotyledons in case of dicot embryo, it occurs laterally, right? It occurs laterally. The position of cotyledons we have seen in this structure, they are present at the lateral side, right? And here you will see a single cotyledon occupies terminal position. Here the position is terminal and here the position is lateral. Now the next difference is coleoptile, right? Coleoptile absent in case of dicot embryo, and in case of monocot embryo envelope of plumule is called the coleoptile. जो उसकी कवर होगी प्लूम्यूल की उसको क्या कहते हैं हम लोग? कोलेप्टाइल. लेकिन वो एब्सेंट हो किसमें? डाइकॉट एम्ब्रियो में वो एब्सेंट होगी. कोलोराइजा, it is absent in the dicot embryo, right? It is absent in the dicot embryo, but in case of monocot embryo there is a protective sheath of radicle, right? There is a protective covering of radicle. Here scutellum, sealed, this sealed कहते हैं scutellum, it is absent in the dicot embryo, and here in case of monocot embryo it is present, right? So these are the differences between a monocot and a dicot embryo. Clear? चलिए. Now the next topic is seed. We have already discussed in our chapter what is microsporogenesis, what is megasporogenesis, what are the events in microsporogenesis, what are the events in megasporogenesis, what are the agents that is responsible for pollination. Then we have discussed about the pollen pistil interaction. After that, what we have discussed? Double fertilization, right? So what would be the ultimate result? Why we are studying, right? So we will look at here pre-fertilization. Pre-fertilization means before fertilization, the product is it is the egg, right? It is the egg is present here, right? After post-fertilization, it will convert into zygote. Clear? These are the important points, please learn them, right? With pre-fertilization, egg is there, right? And after post-fertilization, zygote will form. Now second is your secondary nucleus, right? Secondary nucleus means your n plus n condition. It will form PEN, primary endosperm nucleus. Endosperm का जो formation है, right? Endosperm जिसे बनेगा. Then third is your remains of nucellus, right? What will they form? They will form perisperm, right? This is the perisperm they will form, and the example is your black pepper. Now synergids and antipodals, they exist before pre-fertilization, but post-fertilization they do not exist. Clear? Ovule, here you will see it here, seed final production of sexual reproduction. So before it is a ovule, and after post fertilization it converted into the seed, and it is the final product of sexual reproduction. Now the micropyle, right? The micropyle it will convert in micropyle of ovule, micropyle opening, right? Micropyle is the opening, so the micropyle of the seed, it will convert into the micropyle of the seed, and here the entry of the pollen tube, right? It is also where place where the entry of the pollen tube will occurs. Now the next is outer integument, right? Outer integument, it will convert into outer seed coat testa. Outer integument, it will convert into outer seed coat, that is called as the testa. Inner integument, it will convert into inner seed coat, that is tegmen, right? This is the name, tegmen, tegmen, right? We have studied this structure, outer integument, inner integument, ovule का जो structure पढ़ाया था, outer integument, inner integument, clear? Now the ovary, it consists of many ovules, and the final product of it is the fruit. The fruit will form, right? So ovary wall, what the ovary wall will convert? It will convert into fruit wall pericarp, clear? So it will convert into fruit wall, that is called as the pericarp. Okay, I hope now it is clear. These are the important points, right? You must learn them, right? We will try to recall it again. We will try to learn what before fertilization, egg is present, right? Egg is there, it will convert into the zygote. Zygote is your diploid 2n, right? Here secondary nucleus, it will convert into PEN, right? PEN, endosperm, triploid primary endosperm. Remains of nucellus, right? Nucellus is your diploid, याद आया? Nucellus is your nourishment के लिए था nucellus. So the remains of nucellus, it will convert into perisperm, right? And the example is your black pepper. Black pepper is the example. Synergids and antipodals, where we have learned in the embryo sac, right? So synergids, antipodals, in the pre-fertilization event they are present, but after the post-fertilization they do not exist. Ovule, right? Ovule, so here the ovule, it will convert into the seed, and seed is the final product of sexual reproduction, right? Seed is the final product of sexual reproduction. Now opening micropyle of the ovule, right? Micropyle of ovule, it will convert into micropyle of the seed, right? So micropyle of the ovule will convert into the micropyle of the seed. Outer integument, right? Outer integument and the outer integument, it will form the outer seed coat testa, right? Outer seed coat testa, and the inner integument, it will form the inner seed coat, that is tegmen. Ovary, it consists of many ovules, so the ovary consists of your many ovules, and it will form the fruit, right? Ovary will form the fruit, and the ovary wall, it will convert into fruit wall, that is called as the pericarp, right? The fruit wall, and the fruit wall is called the pericarp, right? So now we are moving to the next topic, that is your seed. So seed, we are discussing in angiosperms, seed is the final product of sexual reproduction, right? It is often described as fertilized ovule, right? Seed को क्या कहते हैं हम लोग? Fertilized ovule भी कहते हैं, right? Seed को हम क्या कहते हैं हम लोग? Fertilized ovules. So they are seeds are formed inside the fruits, right? Seeds कहाँ form होंगे? Fruits के अंदर. And seed typically consist of what they consist, क्या क्या होगा उसके अंदर? Seed coat, right? Seed coat, cotyledons, right? Seed coat, cotyledons, and then embryo axis, right? Embryo axis, right? So cotyledons, what are cotyledons? Cotyledons are of the embryo, right? They are the simple structures. So we have learned in monocotyledons and in the dicotyledons. So the cotyledons of the embryo are simple structures. Clear? Generally thick. So cotyledons are generally thick and swollen due to the storage of food reserves. So the food reserve is present there. So the food reserves is present there. So they are generally thick and swollen, as in case of legumes, right? As in case of legumes. So now when we are talking about the seeds, it may be categorized into two categories, right? There are two categories of the seeds. One is your non-albuminous or X-albuminous, right? Non-albuminous or X-albuminous, and second is your albuminous, right? Second is your albuminous, or other name is X-albuminous, right? So here you will see in case of non-albuminous there is no residual endosperm. जो एंडोस्पर्म है वो बचेगा ही नहीं, पूरा कंज्यूम हो जाएगा. Completely consumed, it is completely consumed during embryo development, right? Embryo development के दौरान क्या होगा, ये पूरा completely consume हो जाएगा. क्या एग्जाम्पल है इसका? पी के केस में होगा या फिर ग्राउंडनट के केस में होगा. इन केस ऑफ पी एज वेल एज इन केस ऑफ ग्राउंडनट. Here you will see retains a part of the endosperm as it is not completely used up during embryo development. जो एल्ब्यूमिनस होगा, ठीक है, उसमें कुछ पार्ट एंडोस्पर्म का रिटेन रहेगा, कंप्लीटली कंज्यूम नहीं होगा एम्ब्रियो डेवलपमेंट के दौरान. क्या एग्जांपल है इसमें? वीट का, ठीक है ना, वीट, मेज, बार्ली, कैस्टर, राइट. सो दीज आर द एग्जांपल्स, राइट. आई विल शो यू द स्ट्रक्चर. I will show you the structure from the book. You will look at here. Right, this is the seed coat. Right, this is the seed coat. This is the endosperm part. So endosperm is persist here. Right, this is your cotyledon. Right, this is the this is the cotyledons. Here you will see it here, albumin endospermic. Right, so it is not completely consumed. This is the hypocotyl axis, right? This is the hypocotyl axis, and you will see it here. This is the shoot apical meristem, right? This is the root tip. This is the non-albuminous, right? Non-albuminous or non. Here you will see the endosperm is completely consumed. Clear? So what we have learnt in seed, we will recall it, right? In seed. So seed, it is the fertilized ovule, right? Seed is called as the fertilized ovule. It consists of seed coat, right? It consists of your seed coat, cotyledons, and an embryo axis, right? So cotyledons is responsible for storing the food, right? So cotyledons of the embryo are simple structures. These are the simple structures, right? These are known as cotyledons. Generally they are thick and swollen due to the storage of food reserves, right? As in legumes, जो pulses वगैरह जो होंगे, ठीक है ना. फिर जो मैच्योर सीड है उसकी दो कैटेगरी है यहाँ पर. कौन कौन सी है? एक तो है आपकी नॉन एल्ब्यूमिनस सीड्स और दूसरी कौन सी है? एल्ब्यूमिनस सीड्स, राइट. सो जो नॉन एल्ब्यूमिनस है, क्या होगा उसके अंदर? देयर इज नो रेसिडुअल एंडोस्पर्म. अभी हमने डायग्राम में भी देखा था, ठीक है. देयर इज नो रेसिडुअल एंडोस्पर्म एंड इट इज कंप्लीटली कंज्यूम्ड. एम्ब्रियो डेवलपमेंट के दौरान क्या होगा, क्या होगी? वो कंप्लीटली कंज्यूम हो जाएगी. क्या इसका एग्जाम्पल है? यहाँ पर पी है, ग्राउंडनट है. दीज आर द एग्जाम्पल्स, right? These are the examples of the non-albuminous seeds. Here in case of albuminous seed, they retain a part of the endosperm as it is not completely used up during embryo development. ठीक है, इसमें सारा पार्ट कंज्यूम नहीं होगा एम्ब्रियो डेवलपमेंट के दौरान. क्लियर? सो द एग्जाम्पल इन केस ऑफ albuminous is your wheat, maize, barley and the castor. I hope now it is clear. Now we can easily differentiate between a albuminous seed and a non-albuminous seed, right? Now we can easily differentiate them. Now we can, sometimes the question will come, differentiate between a non-albuminous and a albuminous seeds. Clear?